Hi everyone, my name is Chinakaho Malisa. I am a training coordinator with Andrea Guarracino and Ilwazi Open Data Science Platform. As I said earlier, welcome to our webinar series where we are looking at the application of pangenome reference graphs. Today we have two speakers. We have Andrea Guarracino, who will be talking to us about pangenome-based structure deconvolution of the amylase locus. And then later on, we will have Flavia Villani, who will be speaking to us about the enhancing complex trait mapping in recombinant inbred rodent strains through pangenome analysis. To kick us off, I'm just going to read Andrea's bio and then I'll read Flavia's bio when she starts her presentation in about 30 minutes time. So Andrea is a computer engineer who qualified for his bachelor degree and moved on to work for a software company where he has 6 years experience. And that's where he met with biology by chance and fell in love with it and decided to pursue a master's in bioinformatics and then a PhD in the same field. During his doctorate, he explored chromosomal instability in colorectal cancer stem cells for the development of novel therapeutic approaches. In the middle of his research, he encountered pangenomics and began actively working on developing new tools for building and analyzing pangenomes. With that said, I'm going to hand over to Andrea. Thank you.
Okay, thank you too. Hi, everyone. Today, I will show you how you can use pangenomes in the form of pangenome graphs made with PGGB to study the structural configurations of genome loci. I will show you how we applied our methods in the amylase locus, a locus very important for our diet. I will focus more on the method, but I will give you first a little bit of introduction. So about the amylase. As mammals, we digest the starches, these long polymers, into simpler sugars by the amylase enzyme. In human, this exists as a gene family where we have the AMY1 gene that is expressed only in the salivary glands, and the other two genes, highlighted in green and yellow, that are expressed only in the pancreas. On the top, you can see the configuration of this locus in the human reference genome, where we have 3 copies of AMY1 in red, 2 copies of AMY2A, and 1 copy of AMY2B. This to highlight that this locus shows structural variation in the human population, in particular copy number variation. Having a human pangenome, we were able to study this copy number diversity in the human population, and we identified 28 different unique structural configurations that you can see to the left. To study this pangenome, we built 2 kinds of graphs. The first one is the mapping graph. This was built by one of our collaborators, Jason Chin. This graph captures large-scale sequence relationships between the haplotypes and allows you to identify what they are called the principal bundles, that are these colors here. Principal bundles are the repeat units that we can find in this locus. Each bundle has a specific color, and we used this color to produce the visualization to the left. So we are coloring these repeat units in this locus. You can think of them like the segmentally duplicated sequences that you can find in this locus. And then we also built a PGGB graph to study the sequence relationships between these haplotypes in this locus, we were able to build this sequence-based tree to the left, and we identified the 11 consensus clusters. So these 28 configurations cluster into 11 clusters. For example, on the top, this H5 cluster, we have all the haplotypes that have 5 copies of AMY1, 5 copies of 5 red arrows. H4, H2 means 4 copies of AMY1 and 2 copies of AMY2A. So the name of the cluster tells us how many copies I have of AMY1, AMY2A, AMY2B. On the bottom, you can see the ancestral state with 1 copy of each of these 3 kind of genes. But of course, when you study something, you always want more. With the human pangenome, we were able to study the copy number variation in this locus in almost 100 haplotypes. But we would like to have a way to infer these structural configurations into the thousands of short-read samples that you can find online. And that's why we developed a method to exploit the pangenome in the form of the pangenome graph to genotype read samples. To introduce this concept, these methods, I need to introduce a way that we can use to represent pangenome graphs. On the top, we have an example of a little variation graph. To remind you, a variation graph is a kind of graph where sequences, in this example, these 2 sequences, are represented as paths through the graph. So the blue path to represent genome 1, let's go through nodes 1, 2. Let me activate the pointer. So genome 1 is represented as a path through nodes 1, 2, and 4. Genome 2 is represented as a path that goes through nodes 1, 3, and 4. That's a very little simple variation graph. That's the kind of graph that we produce with PGGB. So if we take this graph, all the 4 nodes, we put the nodes in 1 dimension, we can build a path coverage matrix. This matrix has, for each sequence, a row, and for each node, a column. And the values here indicate how many, for example, genome 1, how many times genome 1 goes through nodes 1, how many times genome 1 goes through nodes 2, and so on. So for each cell, how many times each path goes through each node. In this example, this path goes through nodes 1, 2, and 4. So 1, 1, 1, and that 0 to indicate that it does to go through node 3. You can have also a number higher than 1. It's not a binary matrix. These are integers. But if you have also, for example, a short read sample, and you map the short reads against the graph, you could count how many reads go map on each node. You can produce a sample coverage matrix. If your sample has roughly 30x coverage, this is an example of what you could get. So sample 1, 29 reads map on node 1, 31 reads map on node 2, and so on. So you count how many reads for each sample, how many reads map on each node. So we have these 2 kind of matrices. And our methods to genotype samples is based on these 2 representations, on these 2 matrices. With them, we developed COSI-GT, cosine similarity-based genotyper. This pipeline was developed by our collaborators, Davide Bolognini and Alessandro Laviane, that developed and tested this pipeline. And on the bottom, you can see the GitHub repository where this workflow is published. There are several steps on this workflow. The first one is to build our region of interest specific graph. For the amylase, we build the amylase specific pangenome graph. For doing that, conceptually, you have, for example, a target genome, your reference genome that is annotated. You identify a region of interest, the amylase locus, in black in this visualization. You want a way to project the region of interest in all the other genomes that you have that might be not annotated. For doing that, we are using the implicit graph model EMPG toolkit. That's a new toolkit that we're using to better work on graphs, but in the alignment space. So we are working not in the or with a validation graph, but we are doing this by working on the alignments, only on the alignments. So we use this toolkit to project the region of interest on your target genome or your reference genome across all the other genomes. And then we take all these regions of interest on all the genomes that you have, and we use PGGB to build our specific pangenome graph. So here we build our amylase specific graph. First step. The second step, we have this specific graph. We want to produce the path coverage matrix. We take our graph and we can apply ODGI to obtain this representation, where again, each row is a path, so an haplotype, a genome. Each column is a node, and each value indicates how many times this path goes through this node. So you can see here, this path goes twice on these nodes. When there is white, it means 0 times. So we build a specific graph. We build a path coverage matrix of this graph. And now we have to build the sample coverage matrix. So we have to map the samples that we want to genotype against our specific graph. So conceptually, you take your reads, sequenced reads, and you apply a tool to map against the graph. But that's not what we do in this pipeline. We don't go directly from reads to graph. To keep the workflow simpler, we do something else similar. We take our reads, we map those reads against the pangenome, not against the pangenome graphs. So literally, we map the reads against the multifast A. So or your pangenome in the text format, very simply. And next, we project this alignment back into the graph by using another tool that is called GFA inject. So you map the reads against the pangenome and inject the pangenome alignments into the graph. And then you will have again all your reads mapped on your on the graph nodes. And then we use another tool, GraphPack, to go from these pangenome graph alignments to these sample coverage vectors. Here, each vector, each row is a sample. Your, for example, a short read sample. Each column is a node, and each value indicates how many reads map on each node. So we have our specific graph, we build the path coverage matrix, we build the sample coverage matrix, and then we can execute the real genotyping. This final step is called haplotype deconvolution. That to summarize this step in one sentence, the aim here is to find the best combination of n path coverage vectors. N in our case is 2 because humans are diploid. So the aim is to find the combination of 2 path coverage vectors that better represent what we see in the sample coverage vectors. I will show you an example to explain. So we have 3 path coverage vectors representing chi genomes, 3 haplotypes. And we have one sample that we want to genotype. So we want to understand in this sample, the structural configuration. In our case, we are working on the Amylase graph. We want to understand this sample, which kind of structural configuration has. What do? We take all the haplotypes that we have in our pangenome, and we make all possible combinations of 2, both homozygous and heterozygous combinations. Here I'm showing just 3 kind of combinations. What we do, we just sum these coverage vectors each time. And then we compare our sample coverage vector against all possible combinations. For the comparison, we use the cosine similarity. Cosine similarity means that we compute the angle between 2 hyperdimensional vectors. This plot here is an example of what you compute in 2 dimensions, just the angle between these 2 vectors. So we are computing this cosine similarity against our sample coverage vector. That, to remind you, this vector indicates how many reads map against each node. So it's information about your short read sample. We are comparing this vector against this vector here, that are the sum of 2 path coverage vectors. That is information that is in the pangenome. So we are comparing sample information, pangenome information. And the final step is to pick the combination that corresponds to the highest similarity. So in our case, if we pick this combination, we are saying, in this sample, these reads originate from a genome that has these 2 haplotypes in the locus that we are investigating. So for me, this sample in this locus is H1H2. That's the genotype, the diploid genotype. I will show you also what you can do, a few results of the application of this method when applied to the amylase locus. By applying this genotyper to thousands of short read samples, we were able to compute allele frequencies of these complex structural configurations. And we were able to see which kind of configurations, structural configurations, are enriched in agriculture-based samples from agriculture-based population with respect to the other haplotypes that are enriched in fishing, hunting, pastoralism-based populations. It was already known that agriculture was triggering selection for a higher number of AMY genes or longer haplotypes, this to digest starches. But now we are able to see which kind of structural configurations are enriched, not just how many genes we have, but how they are, the configuration that they have in this locus. We also applied this method to ancient European genomes to study the evolution in this locus. And here to the right, I'm reporting 6 configurations are the ones with the highest frequency in these ancient samples. And to the left, there is a plot of the allele frequencies, of the frequencies of these configurations over time, over the last 12K years, 12,000 years before present. And you can see, for example, the ancestral state, the shortest one, so 1 copy for each gene, 1 red, 1 green, and 1 orange. This one, H1A, its frequency decreased over time. The same for the H2A0, so another short configuration. While the configuration with the higher number of genes, so longer haplotypes, increased in frequency over time. And if you make the first two haplotypes first group, for me, this is the ancestral allele. The other four haplotypes, this is the duplicated allele. So you are making two groups. Ancestral allele, duplicated allele. If you apply selection analysis on these two alleles, you would get a selection coefficient of 0.002 that is equivalent of what you will get by applying the same kind of analysis in the lactase locus. And this highlighted for the first time that there is a recent positive selection in this locus. Final slide. This is to highlight the main leaders of this work. You can find way more details on the biology, analysis, and the results in this publication that was recently published just last week. Thank you very much. If you have any questions, feel free to ask.
Thank you, Andrea. To our participants, you can use the chat box or raise your hand to ask a question. I think we can do about 8 minutes of questions before we move on to Flavia's talk. So the floor is open for anyone who might want to ask questions.
Maybe an information that is important, because I remember during the workshop last year that people like workflow managers. This pipeline is implemented in SnakeMake. So Davide, the left here, is using SnakeMake to wrap all these steps that I showed you before. All these steps, these 4 steps, into a SnakeMake workflow. So I suppose it's important. I remember people appreciated NS Core, SnakeMake. So just to give you a detail about how to install and run this genotyper on the cluster, might be an important information.
I see Sean has his hand raised. Sean, do you want to go ahead and ask your question?
Yeah, thanks, Shinakawa. Thanks, Andrea. That's great talk. Yeah, hi. Yeah, so you partly answered the question I was going to ask, is the application side of it. But the other interesting thing I thought about is obviously this sort of method is going to be heavily influenced by the haplotype representation in the pangenome graph. Because obviously the counts on the node.
Yes, you can genotype only what you already have in your pangenome, correct?
Yeah. So if you have, say, for example, a haplotype that's not represented there, it's going to not. I guess with amylase, this was a good example where we were likely to see most of the haplotype represented.
I remember Peter Sutman kind of estimated that our pangenome was already representing 99-something% of what you would expect. So it's a good representation, but you could do something else, something more. You could say, okay, for me, this pangenome, sorry, this one on the left, is not representing everything I would expect in this locus. But if you study this configuration here, you will find details in the paper. You could understand how these different structural configurations are evolving by deletion, duplication. So you could simulate, accelerate evolution and create synthetic haplotypes. So you could create on your, in machine learning with synthetic data.
Yeah.
So you create synthetic haplotypes and perform genotyping again.
Yeah.
Understanding how evolution is working here.
Yeah. It's sort of linked to the other question I was going to, the point I was going to, maybe it's in the paper already, but Did you guys look at the primate data for amylase? I'm assuming
not in this work, but we are in the beginning of applying similar concept on primate pangenomes.
Ah, okay, yeah. Because I guess if we have that baseline, it would also.
But in primates, we don't observe this selection, so probably
Yeah.
we can see something interesting in this specific locus.
Yeah. Okay, great. Thanks.
Thank you.
Chris, you can go ahead.
That was awesome talk. It kind of came in a little late, unfortunately, so because I had a little bit of a conflict, but it may have been answered. So apologies ahead of time. One of the questions I had, because you mentioned that you could pretty much do this with you have to have an intact pangenome, pretty much, right? The graph. So if you had, let's say, short read data or long read data that you had from a new sample and you wanted to see what structure it might fall into based on the graph that you currently have, you don't have a capability yet of doing that, right? So let's say a new set of samples from a population that maybe has some representation in the graph. So if you want to align the reads and say, what's the probability it has a specific structure, can you currently do that?
Just a moment. When you have in my talk, for example, I said several times short read sample, but you can also align a longer read sample. When you align your sample, your sequencing reads against the graph, the method will give you the diploid and aneuploid configurations that better represent your sample. So that's the best that the graph can give you that better represent your sample.
Okay.
So I think it's already doing what you asked, if I got the question. So you have short reads, long reads, you map against the graph, you will get the best representation for each sample.
Okay, so you don't have to go through and do an assembly, redo a graph, or add it to the graph.
You already have a pangenome, and you build a region of interest specific graph, and that's stable. And then you have short reads, long reads. You just have to map against the pangenome.
Awesome. That answers it. Thanks.
Okay.
Okay, I think we will move on to Flavia's presentation. Thank you so much, Andrea.
Welcome.
If anyone has questions for Andrea, maybe we'll have time later on after Flavia's presentation. Flavia Villani will be talking to us about enhancing complex trait mapping in recombinant inbred rodent strains through pangenome analysis. Flavia obtained a bachelor degree in biological science and a master's degree in medical biotechnology from the University of Naples, Federico II. During her master's degree she obtained in 2020, she started working with pangenome graph in the application sense. Currently, she is a PhD candidate at the University of Tennessee Health Science Center. She is building the pangenome model of organisms, inbred mice and rats, using a combination of second and third generation sequence data forward to do transfer for discovery in humans. Flavia, I'll hand over to you. Thank you.
Thank you. So hello everyone. Today I want to explain to you a little what I am doing using inbred rodent strains and what we can do using in particular pangenome graphs. So I think, and you already know why do we need a pangenome. So as you know, the idea is that with the pangenomic approach compared to genomic approach, we are not related to a single reference genome, but what we done is mapped all strains against each other to capture all possible variation that we have, and in particular structural variation. So I want to explain to you what I done using rats inbred strain. So here is an example of the family that I'm using to do some application. And this is the idea. So we have two parental strain. They crossed these two parental strain to have a inbred family of rats. Obviously, the question is that why are you using this inbred family? And the answer is that because we have a lot of quantitative trait already mapped. So with the pangenome, the aim is to improve the genotype-phenotype association studies. So here, what I am showing to you, I'm using PGGB to build pangenomes for all chromosomes. Here, I'm showing to you only one chromosome, chromosome 12. And you can see here that each different line corresponds to a different sample. Here on the top, you can see the reference genome, and you can see that vertical lines correspond to variations. And what I want to show to you is that the length of the pangenome graph is a measure compared to the real length of the chromosome 12. This happens because with the pangenome, we are able to see and to capture all the diversity that we have and a lot of new variations. So what I've done is focalize on the small genetic variants first and then on the structural variation. So here you can see what I saw. So with the pangenomic and genomic approach, I saw two different kinds of variation. So with the pangenomic approach, you can see that I am able to capture also complex variation. For complex variation, I mean a combination of variation, for example, SNP indels or MNP indels and so on. So this is a change that is related not to a change of a single allele. And with the pangenome, we are also able to capture more variants, simple variants. And what I try to show to you is that we done the annotation of these variants, and we saw that we have some annotation here. You can see the impact of these variants, and you can see that we have also a lot of annotation for complex variants. Obviously, what we done before proceed to validate some new variation captured by the pangenome is to see if the variants captured by the pangenome are present also in the genomic standard approach, and this is what we expected. And what I've done is divided the genome in two regions, the easy regions and the hard regions. For the easy region, I mean a region in which I removed all the complexity, for example, a lot of large repeats, or the centromeric region that we don't know very much, but this is what we have from the repeat masker. And what you can see here is that when I done this comparison, in particular in the easy region, and for SNP, the precision and recall and F measure is pretty close to 100%. Obviously, for the indels, the situation is worse, but this happens because indels, you know, are difficult to identify also with the standard genomic approach. And after that, what I've done is see only the variants called by the pangenomic approach, and that are present in repetitive regions supported by the original reads, and they validated some variants with the Sanger sequencing. This is what I showed to you here. So these are the SNPs that we discovered supported by original reads. These are near repetitive or inside the repetitive regions. These are the variants that we validated with Sanger sequencing. Obviously, we done some on the variants that we discovered, and we used the FEWOS analysis that is different compared to GWAS analysis, because in the FEWOS we related the one genotype to many phenotypes. So we see if we find some association between each different variants to many phenotypes. And what we discovered is that two variants are related with some interesting phenotypes. This is the first variant that we discovered is related with blood chemistry glucose and in a significantly way. And here you can see that rats that carrying each allele have a significantly higher blood glucose concentration. And we find that there is also another variant that is located near to our discovery that is already mapped and is related with insulin resistance in other 200 different rats. And this is instead other novel variants that is related with two different phenotypes, insulin glucose ratio and chromogranin expression in hippocampus. When I saw this, I was a little upset because obviously we expected a relationship between these variants and two similar phenotypes. But then in literature, I found something interesting. So it seems that rats that have each allele, they have a higher expression of the chromogranin, a lower expression, lower blood insulin concentration. And what we found is that seems that rats that have H original allele have an increase of this level of chromogranin that regulate the catecholamine storage and release, and seems that variation that affecting the chromogranin stimulate the production of TNF alpha that block insulin, and this causes the accumulation of the glucose. So this is what we see here. This is for small variants, but then I was curious to see also what happened with the structural variation using the Pangenome graph and not only. So here you can see that we use four different approaches, 3 assembly-based and 1 Pangenome-based, and these are here some annotation. And what we done is used the variants discovered by at least 2 different tools that you can see here in purples that have high impact these sites, and we validated these variants with the nanopore technology. And what I want to show to you here is that how the pangenome is able to capture and solve also a really complex structural variation. And I'm starting with the 4 example. Here I'm starting with the easiest one. So here you can see the structural one of the structural variants that we validated and we extracted from the Pangenome graph that has these sites. And this following tandem repeats, and this is present in 31 sample. And seems that mutation in this gene have been linked with multiple neurological disorders in humans. This is instead another example. Here you can see this is an insertion present in this number of samples, and you can see that these following line sign elements, and in this gene that seems have been linked to many inflammatory disease. These are 2 easy structural variants, but what we saw that is really nice is, for example, this structural variation. So what you can see here is that this is not a simple structural variation. So you can see that inside this structural variation, we have also a single nucleotide polymorphism. So it seems that 16 samples has this structural variation, and instead 1 sample has this other structural variation. So this structural variation seems the same. What changes the allele, so the single nucleotide polymorphism inside this structural variation, and as well this following sign element. And this one instead is another example, really complex. And so what I want to show to you here is that this structural variation as well following a line element. And what you can see here is that inside this structural variation, there are a single nucleotide polymorphism, but also some repeats. So what you can see here is that, for example, this sample, 1 sample has this configuration. So and this other one seems the same, but inside this structural variation, we don't have a single nucleotide polymorphism. Or instead, these two samples seems that have the same configuration of structural variation, but you can see that there is a change of nucleotide here, and we lost also another variation here. So it means that with the pangenome graph, we are able to see what there is inside the structural variation. So if there are other variants, instead, if we look only the VCF file, obviously we see only that we have a structural variation, but we don't know the configuration of this structural variation in the different samples, in the different haplotypes. And I try to do also some study of structural variation in mouse. So in mouse, we have the same structure of the, this is another inbred family called BXD. And we have 21 million of already quantitative traits present in this database. So this is a pretty cool data set to do some phenotype, genotype association. And what I want to show to you, I built the pangenome for these two chromosomes. And for the chromosome 19, I discovered this structural variation that is a really big structural variation. And this is strain specific. It means that it's present only in the father of this family and in other 48% of the BXD mice. And the other cool thing is that this insertion is in complete linkage with these two other insertion of 4 base pair and 135 per base. And this other thing is that this structural variation follow in a gene that seems to regulate the T cell homeostasis. And we found that in this near to this variation, we have a mapped quantitative traits in gene network that is related to this T cell regulation homeostasis. And this is instead another example of structural variation in another chromosome. What we found here you can see that we discovered 3 different structural variation, and we have this structural variation here that is represented by the pangenome graph here. This fellow in repeats, and this is an insertion, and this is another strain-specific variation. So is present in the mother of this family and not in the father. But obviously, we have some limitation because we used the linked reads technology, and obviously we have also false positive variants. And the other thing is that we are not able to identify a lot of structural variation. And obviously, we used a VCF file, but you know that with the VCF file, we lost something. So what we want to do is to Pangenome-wide association study. And to do that, we have 2 possibilities. Or we need to go back to the VCF file, but you saw and you know that is not the optimal way, because through the VCF file, we lost the advantages that we have we have when we built a pangenome graph. And the other way is go through the matrix of nodes that this is what Andrea explained to you a little. So this is the idea. We have a pangenome graph, so we can go back to the VCF file. This is the structure of the VCF file in which you have the chromosome, the position, and the genotypes for the different individuals, but we lost something. Or we can go through the matrix of node coverage, in which you have how many reads mapped for each different node and for each different sample. And we call it this matrix of nodes as allele zygosity. And this is the pipeline that I am doing for the mouse. So here, this is the idea. So we build a we have the two parental strains assemblies near T to T, and we mapped the short reads against these assemblies. And in the meantime, we built also the pangenome graph. Then we project this pangenome graph into the alignment. In this case, we have the information from the alignment and from the pangenome graph. And what we obtained is a coverage matrix, the allele zygosity matrix. And from this, we done some association studies. So the idea here is that I'm using as example here, we have one mapping already quantitative trait for this gene. This is a measure of this gene expression. And we have, as I told you, 21 millions of mapped quantitative traits. So we use these as controls for our pipeline. And what you can see here is that there is a big association of a variance and this trait on chromosome 1. And this is what we obtained with the pangenome graph. So we are able to reproduce the peak on chromosome 1, but we have also two different really big peak on chromosome 3 and chromosome Y. And when I saw what there is inside, obviously there are a lot of repeats. So for this thing, now we are trying to leverage a little this method to find a way also to try to use this method for identify and manage repeats. So next step will be using this pipeline on this data that we have here in Memphis. We have this number of DNA samples from hospitalized children. So what we want to do is map these exome reads against the HPRC pangenome and do all the analysis that we have. Obviously, we will have a problem because in human, as you know, we don't have a lot of phenotypes as we have in the model organism. So this is my group. So thank you to all for your attention. If you have any question, I am here.
Thank you, Flavia, for your presentation. That was very insightful. I'm going to open the floor for questions from the participants. You can either raise your hand to ask a question or type in the chat box. I don't see any questions. Maybe if there are questions for Andrea as well, now's the opportunity to ask them. We've got about 10 minutes left on the call. And while people think about the questions or comments they might have, I'll just mention that we do have a human pangenome bring your own data workshop coming up in October. So that will be a hands-on bring your own data workshop. It's more for people to apply the pangenome graphs that are available to the data that they have available. So if that applies to you, I invite you to visit the link that I've just pasted in the chat box to see what the workshop is about. There's also an option to apply to the workshop itself. So if you visit that page, it will give you all that information. And then secondly, this recording will be made available on our YouTube channel. It should take about a day or two. By Monday, we should have the videos up on our YouTube channel. Thank you for giving me that opportunity to explain on that. So Mohammed, I saw you had your hand raised. I don't know if you still have a question. You can go ahead.
Yes, please. Thank you, Andrea and Flavia, for the talk. Flavia, I'm asking, which tool are you using to detect the SNPs in the structure variants?
So what I done is obviously started from the VCF file, and then I used Bandage. So this is a tool, maybe we shown to you during the workflow that when we went to South Africa. So the idea is to extract only the position of the structural variation with the OG extract, and then from this I open this with the Bandage, and I saw what there is inside. And then as well, I used the OG to understand how many samples has this structural variation or not. So I used both OG and Bandage. This is what I done.
And it allows you to detect the SNP as well?
What?
You said that you can detect SNPs in structure variants, right? I'm asking, yeah, you use the same tool, the Bandage and OG, to detect the SNPs.
Both together. Now I think Andrea also done a new, faster way to do this with OG. I don't know if you put Andrea inside OG this thing that we done.
I wanted to clarify, the variant calling step is performed in PGGB by using VG deconstruct. That tool will call you SNPs in those structural variants. It's a way to go from your graph in GFA format to variants VCF format. So this is done by VG deconstruct. What Flavia shown you is a way to study those variants into the graph space, because you project your graph into a VCF file. If you want to go back, you take variants, you find the coordinates on the reference or node information, because in the VCF file you will have the column, the ID column of the VCF file contains the boundaries of the bubbles. To remember, variants in the graph are bubbles. You have two nodes where the bubble starts and ends. You can take, for example, those two nodes and ask OG, could you please extract me from the graph this bubble? You do the extraction, and then you use OG or Bandage to visualize and study what's happening in this locus, in this bubble. So VG deconstruct to call variants, and then OG Bandage to go back from the specific variant back to the graph.
So yeah, this is this is the problem, because we need always a VCF file to understand where the variance is located, and then from this you can go, as Andrea told you, use it the start and end of the bubble, or maybe you can use the coordinates and you can add some base pair before and after these structural variants, load it in a Bandage and see what's happened. But obviously we want more, so we want to skip the VCF file, but it will be really a long way to do that.
Yeah, to skip the VCF file, we have, for an example, those matrices that I've shown you also before, Flavia showed you data before, that those matrices are a simplification. We are just projecting the graph in like two-dimensional 2D matrix. But we are trying to see, is it enough for doing GWAS? It seems so. Is it enough? There is a doubt, because in that matrix, we are still not giving the tools the bubble information. When you have a little bubble, you will have first node of the bubble, two nodes for the two SNPs, for example, last node for the bubble. So we have 4 nodes. These two represent the variance. But these two SNPs, for example, have column 2, column 3. And the downstream tool for doing GWAS at the moment doesn't know these two nodes. Are telling something in the same genome position, like a SNP, biallelic SNP, same position. These are 2 different nodes in this matrix. So at the moment, the tool doesn't know that these two nodes represent the same locus. So we are trying to see, is it enough or we have to give also bubble information for the GWAS, for example. That's what we are exploring with Flavia. Yeah, another thing is that we are using a standard method as GEM. I don't know if you know this tool that is done some genotype-phenotype association. So we are trying to adapt the matrix of nodes for this tool. But obviously we can use what we want because it's simple regression analysis. And as I told you, in the GWAS, usually they use the only SNP, simple SNP. So we are trying to see if we are able to use all the complexity of the graph as the repeats. And this is what we are trying to understand and debug.
Yeah, thank you.
I just want to check if there are any more questions from the audience. I see Sean has typed in the chat box that great work, lots of potential for applications. Okay, so I don't think we have any more questions. Thank you for everyone who made the time to attend. And thank you, Flavia and Andrea, for making time to show up. And Takuza, do you want to go ahead and ask your question before we've got about 3 minutes? I'll just give that one to you if you want to ask.
I mistakenly clicked it. Sorry about that. But great session anyway.
Okay, so okay, thanks, Taku. Yeah, so great session. Thank you, Flavia and Andrea, for attending, and thank you everyone else on the audience for coming to the webinar and asking questions as well. I hope you have a good weekend ahead of you. For those who still have a work day ahead of them, all the best with the rest of the day. Thank you, everyone.
You too.